Hello, my name's Sarah Richton. I'm a University of Bristol PhD student who is currently looking at effective management of Livingstone's fruit bats, which are a large fruit bat on the Comoros Islands off the coast of Madagascar. I'm honoured to have been asked to present this bat advent calendar this year for Global South Bats because it gives me an opportunity to talk about my colleague Angelica Menchaca and friend who has authored an amazing paper on conservation units and historical matrilineal structure in the tequila bat. Now, Angelica and I know each other because we were in the same lab together for Angelica's PhD time, and we share the same supervisor, Gareth Jones. And the reason I'd like to talk about Angelica's paper is because she has taught me everything I know really when it comes to genetics. And it's only because of her support and her empowerment that actually I've been able to conduct my own research on genetic diversity and pedigrees in the Livingstone's fruit bats. So I have a huge debt of thanks for Angelica and the amazing work she does. But that said, I am definitely not in the same league as her when it comes to genetics, so I will do my best to summarise her paper as well as I can, but apologies if I get a few things wrong. So again, to reiterate, the paper's name is Conservation Units and Historical Matrilineal Structure in the Tequila Bat. Scientific name, Leptonicterus yerbabuene, I think. Apologies if that's wrong. Lead author, Angelica Menchaca. And then we have Maria Clara Artega, Rodrigo A. Medellin, and Gareth Jones as co-authors. The reason I wanted to look at this paper is because it has really important conservation value. Its application is quite strong. Angelica looked at mitochondrial DNA and control region DNA in several different groups of tequila bat. She sampled across Mexico, out in the field, and she went to three different populations. So one in northern, one in central, and one in southern Mexico. And in these populations, she put up mist nets and she captured 20 bats at a time and collected tissue samples to bring back to the lab. So she also had a fourth population of data that was provided by one of the authors. Her plan was to look at whether or not there are two distinct populations of tequila bat in Mexico that warrant individual and targeted conservation effort. This is because there are distinct differences between these two populations. So in the south, we see a group of bats that tends to be non-migratory and stays in the same location all year round, and then a more northern west group of bats that do migrate quite a long distance throughout the course of the year. So Angelica was adding to this discourse to say, is there a genetic difference in these populations? Are there recognised behavioural differences along with migration that actually warrant them being conserved as individual groups? So once you collected the samples, you took them back to the lab, and I won't go into too much detail about the genetics behind it, but she extracted genomic DNA, processed them with the standard kind of Taq polymerase setup, PCRs all over the place, and then got her data back. She sequenced everything at a lab outside of Bristol. So once she's done all of that, she's looked at mitochondrial haplotype diversity between the different populations and basically had a look to see whether they are the same, whether they are different. So her work in the end shows that actually with both of the mitochondrial markers, so site B and control region, there's a very consistent pattern of genetic diversity across the two populations with high frequency haplotypes common across all colonies. However, the data obtained from the control region sequences does support the presence of two matrilineal mitochondrial lineages, so matrilineal lineages, I guess. So this does add to that discourse to say that these are independent populations, and therefore the conclusion really of this paper, along with the timing of reproduction, along with the migratory behaviour and the habitat requirements, Angelica's work adds to the idea that actually these are two distinct units of bats, tequila bats, and so these variables that have been looked at really should be fed into frameworks like the IUCN Red List criteria to establish these two populations as needing targeted conservation work. So Angelica's work really comes into this and supports this idea, and in the future will hopefully change the way that people conserve this species, which is remarkably important. So Angelica has written a paper that has direct applicable conservation value of high importance. And that's why I wanted to look at this paper, because she has done an awful lot in the field, she's done an awful lot in the lab, and she's come out with a result that really will add to the conservation of this one species. So very impressive. So that's me. I hope you enjoyed. Please do go and seek out the paper if you're interested. Again, it's up on Global Ecology and Conservation, and it's the Conservation Units and Historical Matrilineal Structure in the tequila bat. I hope you enjoy the rest of the Bat Advent calendar and have a lovely Christmas. So thank you very much for listening.